let's get started. And before I introduce today's speaker, Bob Wayne had a quick announcement that he wanted to make.
Just want to remind everyone, the Dynamite seminar, in addition to Lee's seminar tomorrow at noon in Gonda, Hans Hoffman is going to talk about cichlids and how they recognize each other and how they speciate, behavioral mechanisms for speciation. So I really encourage you to go. Hans Hoffman, Gonda, right near the Synapse tomorrow at noon. So please go. He'll be home in our department if the Neurosciences Initiative hires him. So he's a potential member of EEB.
So he's a job candidate.
Candidate, kind of informal. I think it's almost a look-see, but he's on our list anyway. He was willing to consider coming to UCLA. He heads a whole institute at University of Texas, Austin.
Okay, good, thanks. Okay, so if cichlids are your thing, be there tomorrow.
You worked on insects too.
And whatever. Yeah, I had insects. So it's my pleasure to introduce Lee Katz, today's speaker. I'm introducing him in part because Lee is a friend and a colleague who works on invasive species locally and conservation issues associated with that, and in part because, as I'm sure you know, the Kats Center co-sponsors usually one or two speakers a quarter in the EB series, and at least, you know, what I seek to do in that co-sponsorship is to identify people who do great science that really is conservation relevant, and at least for my money, Lee is as good an example of that as anybody I know. His work is great. It's fascinating ecological research, and it's fascinating and important conservation work, especially locally and especially in the Santa Monica Mountains. So Lee received his undergraduate degree at Calvin College, a school I'm not intimately familiar with, in Grand Rapids, Michigan. Went on to get his PhD at the University of Kentucky with Andy C. Those of you who either do behavior, predator-prey interactions, or amphibian work will know of Andy. He's a total rock star in all three of those areas. Lee was one of his very successful PhD students. Andy then moved to UC Davis, and I think actually will be here next week giving a seminar, so that's another one to put on your list. Anyhow, and then in 1990, Lee moved to Pepperdine University as a newly minted assistant professor, and he's been there ever since. And Lee, again, in terms of just, I think, a really interesting sort of professional profile, has been leading a very, very successful and interesting research program, working, you know, primarily with undergraduates at Pepperdine, because that's what Pepperdine is all about. And he's also managed to do that while sort of climbing the administrative ladder at Pepperdine, and I don't want to get this wrong. And he is now vice provost for research and strategic initiatives or innovations at Pepperdine, which means sort of running a lot of the research program and also doing fundraising and development for Pepperdine, while doing his own research, while being a teacher, while advising students, while doing all of that. So Lee's he's quite an amazing guy, and it's my real pleasure to introduce him to talk about the work of his that I think he's best known for, and that I certainly know the best, which is this kind of really fascinating, important work on invasive species, largely crayfish is the system I know the best, and how these invasive crayfishes in the streams in the Santa Monica Mountains have just changed the whole structure of the ecological communities out there. Describing that, and also some interesting stuff on what we can do about it. So,
great. Well, thanks, Brad. I appreciate it, and thanks, Dick, for the invitation. It's great to be here. I was at an event last night where CNN journalist Fareed Zakaria was speaking, and he said he never uses PowerPoint because everybody he says that uses PowerPoint usually have no point and they have no power. So I'm going to try to defy that and at least have 1 point today. So I want to talk about this work that Brad just mentioned. It's local work. It's a local story. I came to Pepperdine in 1990, and I knew we weren't going to have a graduate program to work with students there, and so I began to focus on work that was close by, was easy to get my students to, and interesting scientific problems. And I had worked on streams in the Midwest, streams in the South. I'd worked on predator-prey interactions, and so along those lines, I was looking for topics in that general area. So I should point out that these are my undergraduate colleagues who are going to contribute most to this work. They graduated this past year. And then actually Gary Bucciarelli, who's in your area, started contributing to this work a few years actually before he started in the program here. So he was one of several people that were out in the streams counting frogs and salamanders and eggs, and so I appreciate the contributions of all these folks. I don't really need to go over this other than to say that for, say, the last 15 years or so, there's been a pretty widespread concerted effort to survey streams, biodiversity in streams in the Santa Monica Mountains. And this work is with Seth Riley from the National Parks Service, Rosie Daggett, Robert Fisher from the USGS, state parks folks. We all got together 15 years ago or so and said, hey, let's do this as an effort. So we have a yearly meeting where we kind of highlight what we're going to work on. But every year I take grief because, of course, they wanted to start that 15 years ago, but I had started 20 years ago. And so when we decided to carve up the streams, I just claimed all the cool ones. I just said, you know, I've been in that one, that's mine. I've been in that one, that's mine. And so this is a bitter point with those folks. If you talk to any of them, they're all grouchy because their streams aren't as pristine as the ones that, you know, we've chosen to study, but they need to get over it. So the story is going to be a lot about newts, but it's also about biodiversity of streams here in the mountains. And I always have to put up a picture of one of my favorite animals. If you've been in our local streams, this is a charismatic animal. This is Pseudacris cadaverina, the California tree frog. If you look at the old Stebbins field guides, they said that you don't usually find this frog with Pseudacris regilla, but the truth of the matter is you find them together all the time. But whereas regilla is a habitat generalist, flooded mud puddles, any body of water almost, this animal only will breed in flowing, pretty pristine streams here in the Santa Monica Mountains. This is what I call the one-croak tree frog versus the two-croak tree frog. This animal, the males have a one, sounds like a duck, quack, quack. The other frog, regilla, Hollywood's made famous because you can't go to any movie anywhere in the United States without hearing the Pacific tree frog calling in the background. It ruins many movies for me because we'll be somewhere in the tropics, South America, Africa, and I'll go, oh, it's the Pacific tree frog in the background. How handy that it's there. But it's because, of course, it's been recorded for decades and it's filler music, or at least filler noise in the back. This animal is not well known. Very, very little studied. It kind of jumps on and off the species of special concern. It really has no defense mechanism other than to look at a rock like a rock. It's not toxic, it's not poisonous. Its main defense mechanism is to jump into the water and dive to the bottom. That's not a good adaptation as we get later in the story. For thousands of years, it worked well to jump into the water and dive to the bottom to avoid terrestrial predators. Okay? That doesn't work well today for many reasons. So we count adults of these animals. We do these spring surveys. We look at all the amphibian activity. We do the spring surveys. We count adults. We count tadpoles. Eggs of this animal, very difficult to count because the females lay them one at a time. Very different than regilla, who lays little tiny clutches of eggs. But, and I should mention, just because we all like behavior, these males in the springtime are very territorial. If you get in the stream and you do that syllable croak, quack, quack, the males will orient to you and ultimately will attack. They will jump on you, they'll jump on your face. They're not great at discriminating calls. If they think I'm a frog, there's not been a lot of selection pressure to be very good at it. but it's fun. It's fun to do, and I challenge you to do that. This is the California newt. That's the animal that I know best and that I've studied the most the last few years. Just a little bit of natural history background. These are on ground 90% of the year. Underground, we're not knowing what they're doing here in the Santa Monica Mountains much. They're underground, we don't know what they're doing. However, when the rains come, they begin to migrate to the streams to breed. And this picture is taken underwater. These are some very excited males. The males at this point have a very dark color. Females have a lighter color. She has a swollen belly right here, and if you look closely, a frown. Because these are these mating balls that occur in newts, where the males are very aggressive. In fact, if you know newts at all during this time of the year, if you stick your finger in the water and wiggle it, they'll unplex your finger. They'll unplex a pencil. They are geared up. This is a really cool photo, again underwater, of a female. She's deposited a couple eggs, masses here. She's actually feeding with her forefeet. As they begin to absorb water, she's making sure they're glued onto that stick. And one more is coming out right here. You see a whole bunch of other eggs here. Each white dot is an embryo. There's not a lot known about microsite selection here. What's going on, how the females choose where they want to lay eggs, why this stick, why this pool. I'm going to talk about it a little bit. Do we get a bunch of copycat behavior? You know, we all know organisms do that. If a female's laid here, then she says, hey, that's a good spot, I guess I'll lay there. We don't know. We don't know. It's never been looked at. It's never been experimented. We just don't know. But this is kind of what's going on in our streams in the Santa Monica Mountains, or at least it's supposed to be what's going on in our streams in the Santa Monica Mountains. Here comes the bad news part of the story. This is the red swamp crayfish, Procambrus clarkii. And it is from the Mississippi area, from the Mississippi drainage. And it's now been studied on folks that look at crayfish. It's one of the most aggressive species there is. If you challenge this animal, it'll challenge you back. Yesterday, you know, I had one clamped onto this finger for about 5 minutes. They're not very sensitive about letting go once they're clamped on. This is a big crayfish. It's an aggressive crayfish. And it's a really, really good invading animal. If you think about traits of invaders, right? It's aggressive, it has parental care, it protects its young, it's an omnivore. It's exactly what you don't want to get into our streams. And so these streams that we've been looking at for the last 20 years, I got to tell you that they were surveyed intensively in 1970, that's long before I got here, and there were newts and California tree frogs and Pacific tree frogs in all of them in the 1970s. And when we began looking at this in the 1990s, we began seeing lots of crayfish in some streams and no sign of newts. There are streams in the Santa Monica Mountains. I have not seen one newt in 20 years, but they are recorded there in 1970s. Okay? So that's kind of the puzzle that my students and I have begun trying to figure out, sort out what's going on there. Here's a picture underwater. It's a little cloudy, but you get the gist of what's going on here. This is underwater. This is a very large crayfish who's eyeing up newt egg masses. And sure enough, it turns out it's a favorite food. Now, keep in mind, Gary can tell you a lot about this. These egg masses are very toxic. They have tetrodotoxin. Toxin was first described in Tarica from the egg masses out of the Berkeley group. And yet, it doesn't deter these guys from just going right through them. Almost nothing will eat these things in our Santa Monica Mountains. Occasionally, I'll see a caddisfly larva burrowed in. And I'm not sure if they're actually going for an embryo or the jelly and the gelatin and the other parts of the egg mass. But otherwise, once they're laid, they don't disappear. Okay, they're there and they're easy to count. And so counting these eggs is kind of the main assay that I adopted several years ago to say how good are they doing. Are they getting decent reproduction? Does it look like they'll have treatment this year? Or does it look like it's going to be a bad year for newts? Based on both adults and egg masses. By the way, feel free to throw a question up anytime here. This is interactive, whatever, however you want to do it. I'm happy to answer your questions as we go. Yes, good question. So dating way back to maybe the 1930s or 1940s, or even earlier maybe, they were being brought into California to use as fishing bait. And they are still legally sold as fishing bait. You can go to almost any bait store in the San Fernando Valley and buy these animals. Yes, alive, and release them into any stream that you want. If you've decided, hey, I'm done with them, I don't want to hurt their feelings, I'd really like them to live, hey, I'll let them go in Cold Creek. What would happen here is it would alter Cold Creek probably forever, I predict. So, I'm not a fan of crayfish being sold as fishing bait. But Cal Fish and Game are on record as saying, we think it's too late to change the law. It's not going to help. I disagree with that. Yeah. So I've been told, Brad, that you can help make these changes. So
we had illegal, we introduced tiger salamanders that were coming in as fishing bait, and we managed to change that law. It took 3 or 4 tries, but we were able to do it.
yeah, I'm not a great politician, but I'm ready to learn. Oh, so if you look at this female right here, this is what happens. This is a female that has gone into the water to try to breed, and every notch in her tail is a crayfish attack. Crayfish are grabbing her and ripping chunks of her tail out, ripping toes off. And so crayfish not only will eat the egg masses, and of course the larvae, which we think have very little or no toxin, but they're brutal on the adults. And so, how much of this can it take before that female says, I'm out of here, or before she might actually be killed? This is a California tree frog. And I was surveying the stream. The California tree frog was sitting on the rock. I startled it. It did its defense, dove to the bottom, and within literally 4 seconds, a crayfish had grabbed it and ripped its lower jaw off. So you're looking at basically the top part of that tree frog's head. And I'm not exaggerating. I had about 10 seconds to reach down there and get it. And again, I know it's an anecdote story, but it's not a rare or unusual event. This is what's going on with these animals. They're going to lose. Even the adults here are going to lose. Particularly, it's just annoying as heck when they're using the strategy that has worked so well for them in the past. And now you're diving right into these giant crayfish predators.
And they're eating them, they're not just killing them.
Oh no, it had this chunk of flesh and was eating it. The crayfish did, right? And again, I just want to, while I'm going to talk a lot about newts, this is an ecosystem and a community story here, folks. Almost everything I'm going to talk about, I'm going to say this is what's happening with invasives impacting newts. We could talk about how it's impacting California tree frogs, how it's impacting aquatic insects, how it's impacting all the aquatic life. These are omnivore predators. So I'm giving you a narrow slice of the picture here because I haven't been able to collect the whole picture. But I just want to tell you that this is the two-striped garter snake, species of special concern. It's only highly aquatic. And what do they eat? Here is a two-striped garter snake with a California tree frog tadpole sticking out of its mouth. It's their favorite food. The bottom line is, in the streams where the tadpoles are gone and the frogs are gone, I have 20 years of observations showing that the two-striped garter snakes are just disappearing. They just can't make it. There's no alternative food there. And so this is a more complex story than just, hey, it's bad news for amphibians. I was invited to talk about this in Taiwan in the spring. And while I was on the airplane, literally, while I was on the airplane, I came across this story. And invasive crayfish threatens wildlife in Taipei's ponds. The exact same crayfish. It's the red swamp crayfish. that is decimating the wildlife and the biodiversity in the ponds in Taipei. So it became much clearer to me why they were interested in hearing about this work. Okay, so a little bit about our streams. It's the greatest place to study. You know, I do have work ongoing in Costa Rica and in Argentina, but I got to tell you, this is not a bad place to hang out and study. These are peaceful streams, for the most part, pristine streams, and there's a lot of interesting stuff going on in them. They're also dynamic streams. They change just like California weather. They change quickly, they change abruptly, and in the old days, remember when it used to rain? In the old days, when it rained, we would get, you know, decent flow, even some turbulence. The water would come up quickly, maybe a meter, maybe 2 meters. This is what's supposed to happen in our streams. And that became part of the story, that as the flow changes abruptly and quickly, that it's going to impact the dynamics between these invasives and the natives. So long ago, I'd go out after storms and I'd see things like this. Like, this is a broken crayfish. This is not one that's molted. This is one that, after a nice rainfall, has been bashed up against a rock and brutally murdered. And I could never be happier. And so this is what happens when you have bayou, slow-moving organisms that aren't used to fast-moving mountain streams. How cool. You know, evolution might be on our side after all, right? Let's hope so. So I began to get a little optimistic here. If you poke around some more after a nice heavy rain, you know, I see more. These are not molts, again. These are dead animals. And so I began to get encouraged, thinking, well, this is cool. Something, at least, is hammering these crayfish. Because in this one stream that I'm going to talk about here, it's a unique stream. And that crayfish and the amphibians are co-occurring, and seemingly have been co-occurring for 35 years or more. It's not true in places like in places like Malibu Creek. Malibu Creek, there's too many places to hide if you're a crayfish, too many giant boulders to get under, and you have basically seen the amphibians, particularly California tree frog and the newts, wiped out probably a long, long time ago. But this particular stream, there seems to be some geomorphology here, some flow dynamics that were beginning to make things look interesting. And so if you look at, this is what's going on in Cold Creek, a very nice pristine stream in a preserve. So I've got rainfall, yearly rainfall on this axis. I've got density of Cold Creek Tarica newt egg masses here. And just a 5-year, 6-year snapshot of what happens. You know, when you get little bits of rain, you get some newt productivity. You don't get huge newt migration, but enough come down, lay some eggs. As you get more rain, it's a better year for newts. More newts migrate out of the hills. They lay more eggs. Little rain, you see the pattern. Okay, this is a pattern. So it makes sense. The newt egg masses and the newt activity will fluctuate with rainfall patterns. That's Cold Creek. But then I had these data from some snapshots in the 1990s in Trancas, a different stream that has crayfish, and this was a fun natural experiment. 1997 became an El Nino year. And so if you see, these are the number of pools that I surveyed, total number of crayfish, and total number of newt egg masses, right? And so huge number of crayfish in 1997, 0 newt egg masses. By the way, these adults are there. How do I know they're there? Because I can see them, I can count them. We know they're long-lived. I'm going to tell you here in a little bit that I've got newts that have been pit tagged for over 20 years. And they're still walking around our mountains, and I'm still picking them up and scanning them, and hey, I saw you first 20 years ago. So it tells me they're probably living 30 years or more, right? So I started seeing this pattern and I thought, wait a minute here. Then El Nino comes in, crayfish are down to 1, we get some great newt egg masses. 1 year after El Nino, crayfish start to come back. I'm like, whoa, there's an interesting pattern here. So a year or so ago, I decided to try to piece it all together. And so this is a little bit busy, but it's busy because it's a long-term data set. And we'll get some time here to orient. I know several of you are involved in long-term data sets as well. And I just think we gain so much from these long-term data sets. 2 and 3 year snapshots are very hard to tell what's going on. So the bars, again, blue is rainfall for the year. Eureka, egg masses is the orange here. And the red are the crayfish. So we've got crayfish added that you didn't see on the Cold Creek slide here because there aren't crayfish in Cold Creek, right? So here we're just dealing with rainfall and newts. Now we go to this slide, and I should point out this is average rainfall in Los Angeles, this dotted line right here. Okay, so below average rainfall, there's some decent number of crayfish. Newts barely have anything going on. Again, below average rainfall, crayfish have said, hey, this is awesome. No newt production at all. El Nino happens. Crayfish are washed out to the ocean. Greatest thing ever. Newts have a wonderful year. Because those adults are hanging out. They're like, oh, give me a decent year. The next year, below average rainfall. Crayfish starting to come back. Enough there, got almost no newt activity. Below average rainfall. Crayfish are really back. No newts. Above average rainfall. You see the pattern. Okay? Let's get closer to what's been going on recently here. 2006. Below average rainfall. 2007, below average rainfall. 2008, below average rainfall. 2009, below average rainfall. Let's look at what's happening with the crayfish each year here. Right? Luckily, we had in 2011, and the newt said, you know, this was unbelievable right here. Even though there were crayfish around, they bred and laid eggs like crazy. I should point out that I began to get depressed right here. This year, and I said, when I saw this, 2007, I said, I can't take this anymore. We're going to get every undergraduate we can go. That's what this asterisk means right here. Asterisk in my field mean undergraduates and mass. And so we went out that year and trapped crayfish. We just trapped the heck. We removed about 3,000 crayfish. And what you can see is we got some newt productivity that we hadn't seen much in the last previous 2 years. So, it's a lot of work. Somebody's got to fund it. Students loved it. A whole bunch of volunteers from around LA County said, anytime you need to kill crayfish, I'm in. And so, you know, part of my message is going to be, are there times where we need to manage these things? And by the way, you can imagine, you're way ahead of me here, the climate change implications. The climate change implications for this, and there are many models that are saying we're going to get lower rainfall in Los Angeles. Well, that will be the end of newts in one population. Now, and I say that, I'm not a geneticist, but I know these things aren't moving much. And I know they're not moving much because, like I mentioned, 20 years ago, I began sliding in these little pit tags. Now, when I read the early embryology research of Victor Twitty, Victor Twitty is one of my heroes, except the end. And Victor Twitty, he did a lot of surgery, did crazy stuff with these. And he noted that often, if he did a surgery on a newt, the toxin would bleed internal and it would kill him because it's very contained in the skin. So I thought, well, this pit tagging thing might be a disaster. I've got to make a big puncture wound. Maybe the toxin will bleed internal and it's not going to work. Well, I've never seen any problem with it. And as I mentioned to you, I now have, at Cold Creek, I have 350 newts. They've been carrying pit tags, some of them for 20 years. So this is a Google map shot of Cold Creek, and I just want to give you a feel for, you know, again, we can't radio track these little guys. We have to physically tag them, scan them, and then GPS their spot. But let's take a look. 2008, this particular individual was there in 2008 in the stream in Cold Creek. In 2009, he was in the exact same spot 1 year later. 2010, woo! That's about 30 feet. 2 years later, he's done a huge movement, about 30 feet. Up, 2011, he went about 20 meters upstream. So this is how much these things move from year to year, right? So if we have habitat alteration, if we get invasive species in there, where are they going? Well, they're going right back to where they were the year before. Right. Well, you rarely even see them above ground at all, outside of the spring. So they're just rarely above ground at all. Well, maybe that was anomalous. Let's go to a second individual. Here's one that was in roughly the same part of Cold Creek, 2008, 2009, there he went, 2010, there he was, 2011. Exact. These animals, and by the way, Twitty, he was marking them by cutting their legs off. He said, the toes are too hard, fast to regenerate. I'll have to remove a leg. Twitty marked them by cutting legs off, and they would march kilometers into the mountains. Like 1,000 and 2,000 meters, three-legged newts everywhere. And then they would return. So my point is, they might be moving up into these hills, but these are pretty steep cliffs. As you all know, these are pretty dry mountains. But even if they are moving, they're coming back literally to within 10 meters of where they were the year before, or the year before, or the year before.
Were those all dry years?
No.
11 was El Nino, is that right?
It was a wetter year.
A wetter year.
Yeah. Yep.
There was also the issue of how the rainfall might affect the natives as well.
Sure. Oh, it's a great question. I'd love to talk about it, by the way. These newts, who aren't great swimmers, right? For the most part, salamanders don't like flowing water. They're doing they're taking the leftover freshwater habitat after things dried out here in Southern California. So frankly, they're not great swimmers. They're pretty dorky at it. But they're amazing at getting out of the water the minute the flows start to get up. So you rarely see one washing downstream or injured from a flow. Okay, I'm going to go back to newts and crayfish in a little bit, but I'm do a quick little side jog right here, because many of you know that New Zealand mud snails have become a big part of our system in the last few years. And people are really, really concerned, right? We didn't have New Zealand mud snails in these streams until about 4 years ago. And so my students started making some observations in the field, and their observations I thought were really good. I'm glad these kids are out in the field coming back and saying, hey, Dr. Katz, it seems like I see high density New Zealand mud snails where the water's not moving fast. No, yes. And I see very low densities where the water is moving fast. Well, it turns out, again, like the crayfish, New Zealand mud snails have not evolved with great flow, high flow velocities. And so they designed some flow experiments using a laboratory system with very gentle flow moving, right? They wanted to compare our native streams, how they held up in flow, and our invasive snails. And what you see with even gentle flow in this artificial stream, the invasive snails really get displaced downstream. The native snails actually end up moving upstream a little bit. How about the amount of time they're moving? Where are they moving? Well, the invasives are trying to move positive movement, but the natives do it a lot more. They know that they can move upstream better. If you go to the field and you do field surveys on invasive snails, so mud snails are not present here. So these are the native snails, February, March, October. You see the densities of our native snails go up and down a little bit depending on flow regimes and weather conditions. But if you go to a stream where the mud snails are present, right, you'll see what happens. When it's low flow, there's a little bit of flow. The New Zealand mud snails are relatively low and the natives are hanging in there. Flow picks up a little bit. New Zealand mud snails drop down. The natives are hanging in there. And then by October, this stream, you have almost no flow. The New Zealand's have taken over, and of course, they have wiped out the native snails in terms of space and what's there. So, we're back to flow dynamics again. Something going on with flow, and I'm much more optimistic, by the way, about mud snails than I am about crayfish. I think mud snails, I've been watching some streams here in the Santa Monica Mountains. They're not moving that fast upstream. We're doing some experiments right now. Mud snails don't like newts. I think there could be a really cool story about having native species intact that deter the spread of invasive snails. I think newts intact are going to deter the movement of the invasive snails. And we have some early data suggesting that. And so I think this story is not as depressing as the newt story. And by the way, I recently gave some of this at the LA Natural History Museum, and folks there confirmed all this. Their observations were exactly the same about their watching of New Zealand mud snails in our local streams. Okay, now I realize this is an isolated story, but I think it's a relevant story and it's an important story. So that stream that I've been telling you about, this Trancas that has all those crayfish and has the newts crying like, why do we care? Well, we care in part because this is a species of special concern. There aren't a lot of populations in the Santa Monica Mountains, right? Some of them are already gone. And so there's something going on here that we think is relatively interesting. Well, I refer to this is the Malibu Golf Course, where in the 1940s, this was a Hollywood hunting club. Errol Flynn and all these big names, they would go here hunting. There was a hunting lodge. They created some fishing holes. And so all these years that I studied downstream, this is Temescal Road right here, it drains from the golf course right into Trancas. So I've been studying this for 20 years. I knew where all those invasive crayfish were coming from. That dang golf course. Now, I don't even like golf that much, and I wasn't about to pay a $200 green fee to go sample up there. And every time I contacted the golf course, they would ignore me. I was dying to get up there and see what was going on. And then in January, after 15 years, the golf course had changed hands, and they called and said, hey, we hear you want to help us get rid of invasive species. We're totally on board. We're going to get rid of every invasive species in our ponds and help us do that. I thought I'd won the lottery. I mean, I know I'm a nerdy biologist guy, but to me, I've been pulling for these fish for these snakes and newts for 20 years, thinking they aren't going to have a chance. Well, I want to show you something. Here's a close-up of the golf course. Here's Temescal, the road Temescal. Here's a 2-acre pond, and then there's another pond right here. And so, but I didn't know what was in these ponds. I knew there were crayfish in there. I suspected there were crayfish in there. But this is a fascinating diagram. We got to study this for just a little bit. This is Jake Kirby, one of my students, worked with me on this project. He's now a faculty member at the University of South Dakota. But Jake studied crayfish distributions in the Santa Monica Mountains. And look at what's going on here. Look at these dark lines mean there are crayfish. Anytime you're connected to an urban center. So, right, these streams go all the way up into Westlake and all those artificial lakes in Westlake. Any of these urban streams or connections to urban streams all have crayfish. Look what happens if you don't have connections to urban streams. No crayfish, no crayfish, no crayfish, no crayfish. Oh, this is a weird one. Oh, the golf course is right there. No crayfish, no crayfish, no crayfish, no crayfish. Crayfish, well, that's Topanga, and look, it gets close to the San Fernando Valley, and people run up there and throw crayfish in. So, again, connectivity to the urban centers here. So these are our talk about a valuable resource. It's these isolated streams they're still maintaining excellent biodiversity of insects, aquatic garter snakes, California tree frogs, newts, and we have lost one right here. And it's because of that darn golf course. So I decided to start exploring that golf course. I finally had permission to do it. And again, it's a beautiful place to do biology. And as we began to do some surveying, this is Evan, who is a surfer, and Evan was this project, and he's like, how am I supposed to check these traps and these ponds? You can imagine the organic matter on the bottom of these ponds. And so he said, I'm going to do it with a surfboard. And so everywhere he walked around this golf course, you just seen golfers looking at him. And then he would put it on top of the golf cart, and he'd hold it on the golf cart and driving it around. I loved it. And so this is how he checked traps for us. And you see how the board is being used here in all these reeds. And, you know, one of the first things we found were western pond turtles. And we, frankly, weren't expecting to find western pond turtles up there because I was expecting to find all these invasive species. But we had set out crayfish traps, and these are the kinds of things I was getting in crayfish traps. I was getting just huge numbers of crayfish, huge numbers of invasive catfish. These are invasive bullheads. They're good size, good eating. Bluegill, that if I'd caught them that big when I was on the farm in Indiana, I'd have mounted them on the wall. Here's a bass. You can imagine, by the way, what bass are good at eating. You know, can you imagine what can go into a bass mouth or a big catfish mouth? Baby turtles surely fit in there. Frogs fit in there easily. And so we began, you know, looking at trapping as many of the turtles as we could, and we ended up with 14 adults and not one subadult or anything smaller. And we initially accidentally found out there were turtles there because we had crayfish traps out, and then one day, you know, a western pond turtle showed up in with our crayfish. So we then switched over to, you know, looking at turtles more intensively, and it turns out that there are probably these old turtles hanging out there not getting any reproduction, just like the newts aren't getting any reproduction, because not only will all those fish eat those turtles, but there are papers published where this same invasive crayfish will eat baby turtles as well. So the golf course is totally committed as soon as we get the support of the Army Corps of Engineers to draining those ponds. And we're going to drain every pond up there and remove every crayfish and every fish. And I think it's an exciting opportunity. I think restoration ecology is unbelievably difficult and maybe not all that practical. And by the way, you know, they're trapping like crazy in Malibu Creek. They're trapping crayfish like crazy in Malibu Creek. And I don't want to be discouraging, but, you know, this is Malibu Creek. And again, you're connected to all these urban centers where all those source populations are. So they've removed something like 12,000 crayfish in Malibu Creek, which probably are going to be good for the steelhead and a few other things. But yeah, I think we have a shot at real restoration here if we can get rid of all those source populations. Yeah.
What about the reproductive biology of crayfish? Are you just pulling up the yolk good enough that they drink the pond? Or you know, are there little ones that are running around the mud that are going to get in there?
Yeah, good question. And I haven't done much of this, so we're going to learn as we go, but we brought in some engineers that are really good at draining ponds. We think that pond is actually spring fed, and so they're now analyzing how to actually kind of block it a little bit so we can get some drying. So these are not chimney building crayfish. These are not burrowing crayfish for the most part. But you're right, there are little ones. I will tell you that there are a whole lot of predators just waiting to help with this. My crayfish traps are destroyed nightly by critters out there who are just ripping out the crayfish. So we get this water drained, we get some drying, we get a couple hundred undergraduates out there, we get some time. I think we can be optimistic. You know, I think we got to try it. Yeah.
Camera traps if you were feeling that here.
Yes, I did. And it's just as you suspected, some of the wiliest raccoons ever, who are swimming out to my traps, you know, swimming out. So this, I just have one concluding slide that just, basically says, you know, I think the patterns are going to continue. Some native species are going to just continue to disappear. I have no doubt about it. In some instances, we might want to consider management. I think it worked one year, and I think, you know, our selective management is a good thing. Restoration might be possible. I'm optimistic about this Trancas site. Management and restoration requires funding. Outcomes will depend on stream flow and climate change. You know, of course, climate change. I'm working right now with one of our mathematicians trying to model what would happen at this Trancas population if we can't get the invasives out, and how many years we might have before ultimately the invasives are going to win. And so it's turning into a kind of a neat modeling exercise that we're at the beginning of. This is just to show you how we trapped some of the crayfish that one year. We set up these barriers, and then, you know, undergraduates showing up in mass with crayfish traps, and it worked. Oh, I just wanted to show you this. This is a recent picture. Crayfish are good for something. This is a bullfrog from Malibu Creek who just had a huge crayfish inside. So the crayfish and the bullfrogs, they're buddies. Well, however you want to phrase it. But probably have a little facilitation going on here, one invasive helping another invasive. But none of our local aquatic species are getting anything out of that. Okay, thank you. Great. Yeah.
She said we could not let them track.
Oh. They're using the fish, I'm sorry, they're using the traps like crazy in Malibu Creek with permission from state parks, and there are native fish there. So, yeah. Yeah, so I'm surprised that, yeah, that is not efficient. Yeah. Done.
Are there or were there any native crayfishes donated to crayfish?
No.
Have they considered any biological control of crayfish or crayfish parasite or anything like that?
It's a great question. I throw this out to my undergraduates every year, and recently they have come back. There are some interesting studies on a fungus that is very targeted to crayfish. But I just don't think I'm the right guy to explore that. But I think we should consider things like that. Bob?
Lee, I remember a few years ago, a water pipe burst on top of a Virgin's water pipe burst in flooded Cold Creek, and there was a lot of scouring and concern about the damage. But in light of your results, it seems to suggest that might have been a good thing if there were crayfish there.
Yeah.
And I'm just wondering, Malibu Creek is sort of for flow is controlled by Tapia as the main wastewater treatment plant. And have they ever considered doing a Grand Canyon kind of experiment where they save up water and do periodic floods and try to wash all the crayfish out to sea?
Well, I'm not up on the volume of water that they're putting out, but you know, at some of our highest rainfall years, I would think would dwarf whatever the tapia could put out. And it's just such a diverse habitat that the crayfish have enough refugia that they just can hang in there.
But as I recall, I mean, I thought Malibu Creek was actually dry for part of the year in a natural cycle before the year.
Yes, right now it is. Yeah, for sure.
So if they let it actually dry out, I mean, there'd be no refuge for crayfish.
Well, and so I've only been educated on this one recently, that not, you know, they used to request tapia not release water during dry seasons, right? Because you're disrupting things that we're hoping for low flow. I mean, I remember years ago having some of my experiments just washed out to the ocean in July, thinking, what happened? And they got fined for that. Well, I've just recently come to find out, in this drought that we're in right now, they're now required to release water in this dry year to keep the steelhead alive. So I just discovered that in the last couple weeks. Brett?
So do you know, you've got this nice kind of this correlation between high numbers of views
Yeah.
and low numbers or absence of Eureka egg masses. And do you know if the Eureka, as you know, some amphibians say the poisonous fishes and trout, a frog will come over to a pond and kind of sniff the water,
Yeah.
and then it will just say, no way.
Yeah.
Do you know if the females are doing that, or if they're laying and then just the crayfish are getting egg masses?
I did work with, actually, I worked with one of Malcolm's students many, many years ago, and he worked on that question, and we set up, chambers in the field. Some had a crayfish in the water and some didn't, and definitely the newts spent more time out of water, and we weren't sure if it was chemically mediated or if they physically were detecting exactly. But it definitely, in that experiment, on a very small scale, very controlled, they were like, I'm not staying in here.
So do you think when you see the lactating masses, you think the females are not right?
I think it's both. I do think it's both. Because I know a few are toughing it out and saying, hey, I'm going to run in there, lay some eggs, take my chances. Because there are occasional times when I'm out there several days in a row where I'll see egg masses and then I'll know they're gone. But I think I've also seen females who have ended their migration and are moving back on the land and are still gravid.
At least in those cases, presumably there will be eggs, and at least energetically,
Yes.
that's a little bit better in terms of they're not blowing.
Exactly.
They're not throwing away all that energy for nothing.
Exactly.
They'll lose another year, and
Yeah, good point.
Any other questions?
Actually, not very related to the topic, but I was wondering that it seems they spend a lot of time on water. How the fire, like fire 1997 will affect that?
Well, you'll have to come to Brad's lab meeting tonight, because that's what I'm going to talk about. Yeah, that's right. Really interesting. The 93 fire really changed Cold Creek, for instance. I have pictures of dead newts that were burned up in the fire. But I also, the bigger problem is it so dramatically changes the habitat. The stability of the hillside is gone, and you see how little those newts are moving, and all of a sudden their pools are filled in. And now I've been studying it since the 93 fire, those pools are gone forever. They're not coming back. If we have increase in fire frequency, I'm going to talk a little bit about this in the lab meeting, because I think it's really important. It's definitely going to hit amphibians.
One other question, which is two related questions. First of all, are the newts are just using tools, right? I mean, they're breeding just using tools.
Yeah, there's small amounts of breeding in the other areas, but 90% is in the pools.
And is it also the case that crayfish are just using the tools, or are the crayfish everywhere?
Everywhere.
So, the pools are pretty discreet. There's not that many of them.
Mhm.
They're small.
Right.
So, is there any way to, rather than try to remove them from the whole watershed, use those fences, but I mean, really good fences and exclude crayfish from the areas where they
Yeah.
breed?
I think our study right here showed it could be done. It just takes time and money. I think it can be done. Because it's pretty easy to figure out where the best newt habitat is. Yeah.
And it is doing good production.
Absolutely.
And the rats,
Well, that was partly why I used the size map that I used here. You know, I was trying to keep just basically big crayfish from moving, and everything else I was letting go through. So, I love the idea, though. I think we need to be more proactive.
Yeah, 'cause I mean, even if it would work, for 3 or 4 years, and you could get
Yep.
full reproduction, and water the family, and it falls apart, you know, you can come back in 5 years, and do it again for 5 years, at least, you know, almost kind of like a little temple.
I agree. Particularly when you have such long-lived animals. Yeah, that's awesome.
No.
All right.